Okay, think about this for a second. Have you ever actually stopped to consider the sheer scale of the microscopic universe going on inside you right now? We are talking about 30 trillion individual cells, 30 trillion! And they're all functioning not as some chaotic mob, but as one perfectly synchronized, living, breathing human being. Welcome everyone to today's Masterclass Explainer. I am absolutely thrilled to be your professor today because we are going to decode the actual microscopic machinery that makes that impossible synchronization a reality. We're exploring the cellular messaging system. So grab your notebooks, get focused, and let's unlock the secrets of cellular communication.
Let's dive right in with a dramatic, real-world scenario. Picture an impala out on the African savanna, literally racing for its life to escape a cheetah. In a fraction of a second, the impala's breathing skyrockets, its heart is just pounding, and those leg muscles are pumping furiously. We all know this as the classic fight-or-flight response, right? But how does that actually happen? How on earth do trillions of completely independent cells instantly coordinate such a massive whole-body response? Well, the secret lies in a single chemical messenger called epinephrine, or, as you probably know it, adrenaline. Today, we're going to trace that exact signal from the very moment it's released to the moment those muscles fire.
Here's our quick syllabus for today. We'll cover the foundations of the messaging system, then we'll break down the 3 stages of communication: reception, transduction, and response, and we'll wrap up by looking at apoptosis, which is programmed cell death. Chapter 1. The Cellular Messaging System, the Foundations of Cellular Communication.
Now, before we look at human biology, we really have to respect the ancient evolutionary history here. Over 1 billion years ago, long before anything like multicellular life even existed, chemical signaling was already hard at work. Soil-dwelling bacteria were actually using chemical signals to monitor their local population density. It's a phenomenon known as quorum sensing. Even super simple, single-celled yeast used chemical mating factors to find a partner. Evolution essentially took these brilliant ancient mechanisms and scaled them way up to make complex animals like us possible. So to understand how it works in humans, our roadmap for today comes courtesy of Earl Sutherland. His pioneering research at Vanderbilt University actually earned him a Nobel Prize back in 1971. Sutherland figured out exactly how epinephrine works on liver cells, and he beautifully dissected cellular communication into 3 distinct stages. Stage 1, reception, where the cell detects the signal. Stage 2, transduction, where that signal is converted and relayed. And Stage 3, response, where the cell finally performs the requested activity.
Moving to Chapter 2, Stage 1, Signal Reception. This is how cells actually hear the message.
The bloodstream is an incredibly chaotic, noisy environment. It's just flooded with thousands of different molecules at any given moment. So how does a single cell know which ones to actually listen to? Well, I absolutely love this analogy. Think of it like a wireless router. A router broadcasts its Wi-Fi signal everywhere, right? But only devices with the correct password can actually connect to it. In biology, the signaling molecule, which we call a ligand, acts like a highly specific key. It floats through the extracellular fluid, sure, but it will only bind to a receptor protein that has the exact, perfectly matching shape, like a lock-and key. If the receptor doesn't fit the ligand, the cell just completely ignores the message entirely.
So where exactly do we find these microscopic locks? Basically, 2 main locations. First, we have membrane receptors right on the surface of the cell. These handle water-soluble signals like our fight-or-flight hormone epinephrine, because those molecules are just too large or too highly charged to push across the cell membrane themselves. Major families of these surface locks include GPCRs, or G-protein-coupled receptors, and receptor tyrosine kinases. On the flip side, we have intracellular receptors, which are located deep inside the cytoplasm or the nucleus. These handle hydrophobic signals, like the steroid hormone aldosterone. Because they're hydrophobic, they act like chemical ghosts. They just slip right through the lipid bilayer of the cell membrane. They literally don't even need a door.
Now, understanding these receptor shapes isn't just some abstract academic exercise. It actively saves lives. Let's look at a real-world clinical application. In certain severe forms of breast cancer, tumor cells harbor excessive amounts of a specific receptor tyrosine kinase called HER2. These receptors are essentially jammed in the on position, which causes the cells to divide rapidly and completely out of control. But scientists developed a brilliantly targeted drug called Herceptin. The molecules of this drug are designed to act as the perfect puzzle piece. They specifically bind to those abnormal HER2 receptors. By locking into place, Herceptin physically blocks the growth signals and halts the tumor development. It is just a perfect example of turning microscopic cellular theory into a profound life-saving medicine.
Chapter 3, Stage 2, Signal Transduction. Let's look at the molecular bucket brigade.
Okay, so the signal has successfully reached the cell surface, but now it needs to get deep into the interior to cause an actual physical change. I like to call this stage the molecular bucket brigade. Very often, this relay happens through something called a phosphorylation cascade. Think of the most complex, beautiful Rube Goldberg machine you've ever seen, or just a massive sequence of falling dominoes. When the surface receptor is activated, it triggers a relay molecule, which then activates an enzyme called a protein kinase. This kinase takes a phosphate group and slaps it onto the next kinase in line, activating it. And so on, passing the energy down the line until the signal reaches its ultimate target. It's a spectacular cascading transfer of biological info. And, just as importantly, enzymes called protein phosphatases sweep in right afterward to remove those phosphates, resetting the dominoes so the cell is ready for the next signal.
But proteins aren't the only ones passing buckets here. We also rely heavily on second messengers. These are small, non-protein, water-soluble molecules or ions that diffuse incredibly fast to broadcast the signal throughout the cell. The major players you absolutely need to memorize are cyclic AMP, calcium ions, and inositol triphosphate. Because these molecules are just so tiny compared to those bulky proteins, they act a lot like a flash flood. They surge through the cytosol in a matter of milliseconds, spreading that chemical alarm way faster than the protein dominoes ever could on their own.
And when these second messengers get messed with, the macroscopic results are huge. Take the deadly disease cholera, for example. It takes hold because the cholera bacteria produce a toxin that permanently locks a G protein in its active on position. This triggers a massive, endless surge of the second messenger CAMP, which leads to a catastrophic loss of water and salts from the intestines. But on the flip side, modern pharmaceuticals use this exact same logic to heal us. Viagra actually works by inhibiting the breakdown of a slightly different messenger, CGMP. By preventing its breakdown, it artificially prolongs the cellular signal that relaxes blood vessels. It's wild to think about, but the exact same microscopic chemical pathways dictate both terrifying infectious diseases and modern blockbuster drugs.
Chapter 4, Stage 3, Cellular Response. Action at the cellular level.
All right, the signal has been received, it's been transduced through the cascade, and now action must be taken. What does the cell actually do? Well, the sheer scale of this is summarized by one massive number, 100 million. When that impala is running from the cheetah, a single epinephrine molecule binding to a single receptor on a liver cell triggers a cascade that amplifies the signal at every single microscopic step. By the end of the chain, that one tiny hormone molecule results in the release of 100 million glucose molecules into the bloodstream. It is an exponential geometry of survival. A tiny, almost imperceptible whisper on the outside of the cell becomes an absolute deafening roar on the inside, giving the animal the massive burst of energy it needs to escape.
Now you might be wondering, Professor, a cell is a highly viscous, incredibly crowded place. How do all these relay molecules find each other fast enough? Good question. The answer lies in scaffolding proteins. These are massive relay proteins that act like a physical workbench. They literally hold 3 or 4 different protein kinases together in a neat pre-assembled network. It's essentially hardwiring the cell so the signal doesn't have to just rely on molecules randomly bumping into each other in the fluid. Not only does this drastically increase the speed and efficiency of the response, but it also prevents crosstalk between different signaling pathways. It ensures your body's internal wiring remains beautifully distinct and organized.
Chapter 5, Apoptosis, or Programmed Cell Death, the ultimate integration of pathways.
Everything we've talked about so far has been about how cells integrate signals to live and react. But what happens when multiple signaling pathways integrate to decide a cell has to die? That's apoptosis. The term actually comes from a Greek word referring to leaves falling off a tree. It is a highly controlled, incredibly organized cell suicide. When a cell gets fatally infected, or its DNA is just irreparably damaged, it initiates a pathway that activates enzymes to systematically chop up its own DNA and proteins. The cell shrinks, it forms these little lobes in a fascinating process we call blebbing, and it packages its remnants into tidy little vesicles. Scavenger cells then safely consume these packages. Why go to all this trouble? It's entirely to protect neighboring cells, because if a damaged cell just violently burst open, it would leak destructive digestive enzymes everywhere. So apoptosis is literally the ultimate microscopic sacrifice for the good of the organism.
And here's the thing. Apoptosis isn't just about damage control. It is a fundamental part of how we are sculpted. During the development of the tiny nematode worm C. elegans, exactly 131 cells undergo apoptosis to create the adult form. No more, no less. In humans and mice, our hands and paws actually start out as solid, paddle-like plates of tissue in the womb. Apoptosis is the specific signal that selectively kills the cells between our fingers, eliminating the webbing so we're born with separate, highly functional digits. Furthermore, every single day you are alive, apoptosis is silently happening inside your body, destroying cells that have acquired mutated DNA and actively preventing cancer from taking root before you ever even knew it was there.
So I want to leave you today with this final provocative thought. We know that when apoptosis fails to occur, damaged cells divide uncontrollably, and that results in cancer. And we also know that when apoptosis happens when it shouldn't, perfectly healthy neurons die, leading to degenerative diseases like Alzheimer's and Parkinson's. Therefore, if we can fully master the vocabulary of these exact cellular messaging systems, the GPCRs, the phosphorylation cascades, the scaffolding proteins, could we theoretically rewrite the instructions of our own biology? Could mastering these microscopic pathways hold the key to stopping aging and disease entirely? Class dismissed. Keep questioning, keep studying, and I'll see you in the next explainer.